The G protein coupled receptors are the largest family of membrane receptors found in eukaryotic cells. Many G protein coupled receptors have 7 alpha helical transmembrane domains and a large extracellular ligand binding domain. When a protein ligand binds to the extracellular domain, the receptor undergoes the conformational changes and gets activated. Upon activation, the cytosolic region of the activated receptor then activates a trimeric GTP binding protein called the G protein. G protein comprises of 3 protein subunits called alpha, beta, and gamma. In the absence of the signal, the G protein is inactive and alpha subunit is bound to GTP. G protein binds to activated receptor. This binding then induces a conformational change in the alpha subunit, causing the GTP to be displaced, allowing the cytoplasmic GTP to bind in its place.
The GTP binding causes a further conformational change in trimeric G protein, activating both the alpha subunit and the beta-gamma complex. These two activated components can now activate and regulate target proteins embedded in the plasma membrane. In some cases, the GTP-bound alpha subunit dissociates from the activated beta-gamma complex as shown in the animation, whereas in other cases it remains bound to the beta-gamma complex. The activated target proteins can then transduce the signaling cascade, activating downstream protein components of signaling pathway, ultimately leading to transcriptional regulation of a specific gene. The alpha subunit is then deactivated by hydrolyzing its bound GTP to GDP. This step is regulated by a protein called RGS, or regulator of G protein signaling.
The inactive alpha subunit reassociates with beta-gamma complex and reforms inactive trimeric G protein at the start of the signaling pathway. As long as the receptor is bound to protein ligand, it is activated and can continue to convert inactive G protein to its active form. However, on prolonged stimulation, the receptor becomes insensitive to the bound ligand and gets inactivated. What actually happens is that a kinase protein phosphorylates the cytosolic domain of the activated receptor. The phosphorylated receptor binds to an arrestin protein. This inhibitory protein inactivates the receptor by preventing its interaction with the G protein. The arrestin-bound phosphorylated receptors are either degraded or restored to activate new signaling pathways.
The alpha subunits of G proteins are divided into 3 subfamilies: G-alpha S, G-alpha I, and G-alpha Q. Let's start with G-alpha S. Typically, G-alpha S stimulates adenyl cyclase and increases the level of cyclic AMP, which activates protein kinase A. This leads to the dissociation of catalytic subunit of protein kinase A, which enters the nucleus and phosphorylates cyclic AMP responsive element binding protein CREB, which in turn binds to CREB binding protein, that is CBP. This CBP interacts with various components of basic transcriptional machinery, including RNA polymerase II, data binding proteins, and thus it regulates the expression of great variety of genes. Unlike G-alpha S, G-alpha I subunits primarily inhibit cyclic AMP dependent pathway by inhibiting adenyl cyclase activity and thus decreasing the production of cyclic AMP from ATP, which in turn results in the decreased activity of cyclic AMP dependent protein kinase signaling. Using different downstream signaling components, G-alpha I subunits trigger transcriptional factors which are involved in STAT3, NF-kappa beta, CJUN, and various developmental activities.
G protein containing G-alpha Q family subunit upon activation stimulate phospholipase C, which then hydrolyze PIP2 to produce IP3 and DAG. IP3 causes the release of calcium ions from ER and regulate the cellular activities. The second subunit, DAG or DAG, activates the protein kinase C and controls the inflammatory responses. G-alpha Q is also observed in the transcription regulation through tubby-like protein and stimulate the RAP7 or developmental processes.
Activating and inactivating mutations or genetic variations in numerous human G protein coupled receptors are associated with the disruption of Gpcr function and result in a wide range of disease phenotypes. Mutations altering Gpcr function are usually classified according to the net change in signaling ability. A general decrease in the relevant signaling is termed as loss of function, whereas a general increase is gain of function. Structural alterations provoked by mutation or variations in the genes coding for the GPCRs may lead to misfolding, altered plasma membrane expression of the receptor protein, blocked or biased signaling, and disrupted ligand or tethered agonist binding.
G protein related diseases are characterized by either deficient or excessive G protein signal transmission. Diseases involving a decrease in the production of G proteins include night blindness, where mutation in G protein alpha subunit affect the response of rod cells to light. Pseudohypoparathyroidism, where the genetic loss of G protein alpha subunit results in non-responsiveness to parathyroid hormone. Excessive G protein signaling can arise through either increased signal initiation or defective signal termination. Increased signal initiation occurs in testotoxicosis, where a mutation in the receptor luteinizing hormone can overstimulate G proteins, resulting in excessive production of testosterone. Diseases arising from defective signal termination includes adenomas, in which G protein lose their ability to hydrolyze GTP through mutation, resulting in excessive secretion of growth hormone. McCune-Albright syndrome, where scattered regions of skin hyperpigmentation arise from hyperfunctioning of endocrine glands.